So the disease is induced by the Shiga toxins. And the Shiga toxins, you see here the different toxins that are produced by E. coli. So the enterotoxins, you have the LT, the STA, and the STB enterotoxin. The LT enterotoxin is a very large toxin, complex toxin, composed out of two parts, an A part, the active part, and a B part, the binding part. And that this makes it a very huge molecule, whereas both STA and STB enterotoxins are very small toxins, as you can see here on the molecular weight. And this also makes that these small enterotoxins are weak immunogens, and animals do not really develop immunity against these thermostable enterotoxins. This is not the case for LT. LT is a very strong toxin, but also a strong immunogen. Now, if you look at the Shiga toxin, it resembles a little bit the LT enterotoxin. It has a similar weight, molecular weight, and it's also composed out of an A subunit and a B subunit. And in fact, the B subunit consists of 5 binding sites. This is also the case for the LTB enterotoxin. So it binds to glycolipids on the cell membrane, and this shows you different animal species and humans in which Shiga toxin can have an effect on the mucosa. Just to show you the difference between pigs and ruminants and humans. So the Shiga toxin of the Edema disease strains is a toxin that binds to clobotetraosylceramide. It's a glycan consisting out of 4 sugar molecules that are linked to the lipids of the cell membrane. And that is different from the Shiga toxins that are produced by strains in sheep, cattle, and humans. And that's also the reason why pig strains are not pathogenic for humans and for ruminants. Whereas, in fact, the opposite can be the case, because pigs, they have both receptors, have the GB3 and the GB4, and the GB3 is the one that binds Shiga toxin of the other strains. So GB4 is the receptor, and the receptor is present in the small intestine. So if the strains colonize the small intestine and they produce Shiga toxin can bind to the epithelial cells and can be transported in the animal and cause systemic effects. So the toxin is present in the small intestine. There is some discussion if it's present in the colon. There is a study that says that there is no presence of the receptor in the large intestine, but it surely is present on the vascular endothelium in different tissues. So like you can see here, and it's also present on the kidneys, the tubules in the kidneys.